Thank you so much, Lee, for such a nice introduction. As Lee explained, my research focuses on molecular evolution. I am broadly interested in the evolution of genes and proteins and genomes and how that evolution is shaped by natural selection, by different evolutionary forces. These are some of the topics that I work on, or we work on in our lab. And today I'm going to talk to you about the rates of protein evolution and what factors affect rates of protein evolution. So what are rates of protein evolution? Some proteins evolve slow and some proteins evolve fast. For instance, here we have an example of a very slow evolving protein. This is an alignment comparing human and mouse. Human and mouse diverge approximately 75. Yes.
Can you please share your screen?
Oh, I forgot to share my screen. Sorry.
No worries.
Okay. Okay, now. Thank you. So I was telling you this is the title, and these are some of the topics that we work on in my lab. And today we're going to talk about rates of protein evolution. And I was showing you this slow evolving protein, which hasn't changed at all in 75 million years of evolution. That's how long ago human and mouse diverged approximately. So almost certainly that protein had exactly the same sequence as human and as mouse in the common ancestor of the two, in the most recent common ancestor of the two, but natural selection has prevented any change from fixating. Any mutation, any non-synonymous mutation that might have changed the protein sequence has been removed by purifying selection during these 75 million years of evolution. Here, on the other hand, we have a fast evolving protein in the same amount of time. You see how many amino acids here represented in a white background, they have changed. So why do some proteins evolve slow like this one or others evolve fast like this one? These different rates of evolution is something that was recognized just as soon as scientists started to have enough proteins to compare. And this is from 50 years ago. This year, I actually recently wrote a small comment on this article from a little bit of a historical perspective. And in this article, Dickerson, what he compared is the evolution of three different proteins. Each of these data points here represents a comparison of two species, the protein between two species, and then in the y-axis, we have the millions of years since the two species split, and here we have a measure of how different the two proteins are. And you see how hemoglobin evolved at a certain rate. Fibrinopeptides evolve much faster. They accumulate mutations much faster, whereas cytochrome C evolve much slower. And he tried to provide explanations for these different rates of evolution. Nowadays, we don't infer rates of protein evolution by looking at proteins usually, but we look at the coding sequences, at the codons. And here we have a piece of the coding part of a gene. Imagine it is in mouse, and this could be the same, the homologous sequence in human. And then if on top of that we add the codons, so now we know that each of these, this is a codon, this is another codon, and so on, and the amino acids that it encodes. And then we can identify some differences, some substitutions that have been fixed throughout evolution. And we can distinguish two kinds of such substitutions. For instance, this one here doesn't alter, doesn't change the amino acid, and that's what we call a synonymous substitution, because the protein sequence remains the same. And for the most part, we think as these mutations, also this is another example, as neutral, fitness remains the same for the most part, with some exceptions. Whereas this other substitution is a non-synonymous one. And this is often deleterious, sometimes neutral, and sometimes beneficial. Mutations are often deleterious. When we talk about substitutions, then they are most commonly neutral because the deleterious have been already been removed. Don't make it to substitutions. So once we can think of coding sequences as synonymous and non-synonymous positions, then we can calculate this ratio, the omega ratio, which is dn divided by ds, also known as the dn-ds ratio, as well sometimes referred as Ka-Ks ratio, depends on the paper, usually as well how many sequences you are comparing. And then dn is the number of non-synonymous substitutions. In this case, it would be 1 for this very short protein or this very short coding sequence. It's the number of non-synonymous substitutions divided by the number of non-synonymous sites. And ds is the same, but for synonymous substitutions and synonymous sites. And then this ratio is equal to 1 because dn and ds are broadly the same when there is no natural selection. This is what we would expect in a pseudogene, for instance. It is higher than 1 when there is positive selection. Sometimes the ratio higher than 1, you only see it in a fraction of the codons, not in the whole protein overall. That's pretty hard to observe. And more often, it is lower than 1, which is indicative of negative selection or purifying selection. The smaller this ratio, the more the protein is evolving constrained by purifying selection. In this talk, I'm going to talk about this ratio, omega, as I'm going to refer to it as the rate of protein evolution. Or sometimes in some of the graphs that I'm going to show you, it's going to be dn that we're going to use as indicator of the rate of protein evolution. But they are not exactly the same, but they are highly correlated. In this work, Yang and Gold, they compare for every Arabidopsis thaliana gene, they found the Arabidopsis ligata ortholog, and then they aligned the coding sequences and they calculated Ka, which is the same as dn, and Ks, which is the same as ds, and then they as well obtained the ratio. And you see how this ratio is very variable. It changes a lot from one protein to another. The mode is around 0.1, but you see them, some of them are above 1 and some of them are 0. So again, why do some proteins evolve very slowly, such as the ones in this part of the distribution, whereas others evolve very fast, such as the ones in this part? Why do we care about these rates of evolution? Not only because it's an interesting question, but also because this degree of conservation of different proteins and different parts of the proteins tell us about their function. Usually, functional domains tend to be more constrained and remain more similar across time than the parts of the proteins that don't have such a clear function. If you had to guess where a mutation is going to be more harmful in this protein or in this protein, I would bet that it's going to be more harmful here. These patterns of conservation indicate that any mutation is going to be deleterious in this protein. Whereas here, some mutations are accepted by natural selection, particularly in the areas that have been have changed throughout evolution. So that tells us gives us a hint of whether mutations can be deleterious or even cause disease. So these are the different factors that I'm gonna be talking to you about. These are different factors that we have been studying in our lab, and already next to each of them, I added a symbol that indicate a sign that indicates whether the factor slows down evolution, that's the ones with a minus sign, or accelerates evolution. These are the ones with a plus sign, okay? So let's start with expression level. There is a negative correlation between expression levels of genes and the rates of evolution of the proteins that they encode. And this is referred to as the ER anticorrelation. E for expression level and R for rate of evolution. Okay, so here we have expression level, and in this case we have represented rate of evolution as Dn. But if we did it with omega, it would be pretty much the same. Okay, so we see this very clear negative correlation. For many authors, they have thought of expression level as perhaps the only factor that's important in affecting rates of protein evolution. There was a time in which it was discussed whether other factors, maybe they had only a minor contribution in modulating rates of evolution at best. But we will see how our analysis don't suggest that. Our analysis suggests that other factors as well are important to modulate rates of protein evolution. The reasons for this anticorrelation are not obvious at all, and the mechanisms that have been proposed to explain it are quite complex. One of the hypotheses, one of the most influential ones, is the translational robustness hypothesis, which recognizes that when proteins are translated every once in a while, around 5 every 10,000 codons, they get mistranslated, which means that the wrong amino acid gets incorporated. Now, which is the path that we have here. So most of the time they are correctly translated, but sometimes they are incorrectly translated. This can lead to misfolding, and misfolding, of course, is deleterious. It can cause all kinds of problems. However, sometimes proteins that are that contain translational errors, they can actually fold and function properly, correctly. Okay. The idea is that highly expressed proteins, because there are many copies in the cell, they are under strong selection to be able to fold, even if there are translation errors. And that's what keeps them evolving slowly. They have to maintain this ability to fold even in the presence of translation errors. Here I have it a little bit step by step. These are some of the problems or in terms of fitness cost that misfolding can entail, such as refolding cost, degradation cost, resynthesis cost, and sometimes even aggregation, which is cytotoxic. And then in order to avoid that, proteins, particularly the highly expressed ones, have been under a strong selection to being able to fold and correctly, even in the presence of some translation errors, and then avoid all of these scores. Okay. So this is one of the mechanisms proposed to explain this anticorrelation. Then working with late Patricia Berningson, we hypothesized that secreted proteins might behave differently, because secreted proteins are synthesized. These are the ribosomes attached to the endoplasmic reticulum, and then as soon as they are synthesized, they are moved to they are actually synthesized into the reticulum, the endoplasmic reticulum. And in the endoplasmic reticulum, there are a number of mechanisms that prevent misfolding, or that can help proteins deal with misfolding. So either they are less likely to be misfolded, or if they are misfolded, the mechanisms they're in are more efficient to deal with that. So these fitness costs are expected to be reduced for this set of proteins. And as a result, we expect less of an ER anticorrelation, because these fitness costs, according to the translational robustness hypothesis, are part of the requirements to explain the ER anticorrelation. So if they are removed or reduced, we expect less of an ER anticorrelation. There are other hypotheses that have been proposed to explain the ER anticorrelation. For instance, the misfolding avoidance hypothesis, which doesn't require necessarily translation errors. It simply assumes that proteins can misfold even if they are correctly translated, and that's what we have here, this path that we have here. And then it posits that proteins, particularly the highly expressed ones, are under selection to follow this path and function and fold and function correctly. Another hypothesis is the misinteraction avoidance hypothesis, which recognizes the fact that proteins sometimes engage in unspecific interactions. Sometimes they interact with proteins that they shouldn't. And then, according to this hypothesis, highly expressed proteins would be under strong selection to not misinteract, because this is again deleterious. And then we have other hypotheses that I'm not going to go into right now. But also these two first hypotheses, at least, they would be compatible with our hypothesis of us expecting a weaker ER anticorrelation in expressed proteins. Because as I was saying, misfolding is less likely here, and it's more likely to be effectively dealt with. And in terms of misinteraction, it's also less likely to occur here because it's a contained space, and also secreted proteins end up in the extracellular space, where misinteraction is less likely to cause catastrophic effects than if it happened in the cytosol. Okay, so taking into account any of these three hypotheses that I have explained, the mistranslational robustness hypothesis, the misfolding avoidance hypothesis, or the misinteraction avoidance hypothesis, we would expect less of a correlation, less of an ER anticorrelation in these proteins. And this is what we observe. These are non-secreted proteins, and they exhibit the usual negative correlation between expression levels and rates of evolution. And these are secreted proteins which exhibit in humans no correlation. Okay. This is protein abundance in this case, in the x-axis, for the whole human body. And this is protein abundance for different specific organs or tissues. And in every one of them, we see a negative correlation for non-secreted proteins, and then no correlation or less on of an ER anticorrelation, at least for secreted proteins. So that as well supports our hypothesis. This is the same kind of analysis, but made with messenger RNA abundance data. And these are non-secreted proteins. Each of these lines is a different tissue. And then here we have the correlation coefficient for the non-secreted proteins. It's always negative, or for the secreted proteins, usually most of the time negative, but always less negative or more positive than in the non-secreted set. Okay, so in every single one of these tissues, there is a difference between secreted and non-secreted proteins. And this is data for other species such as C. elegans, Drosophila melanogaster, and so on. And as well, every time we observe less of a negative correlation, or sometimes no correlation, in the case of secreted proteins. So the first conclusion of this presentation is that secreted proteins escape or defy the ER anticorrelation, which is in line with, it supports or is to be expected under the lens of these, some of the hypotheses that have been proposed to explain the ER anticorrelation. So this allows us to understand a little bit better the reasons of this ER anticorrelation. The next step was to look at N-glycosylated proteins. N-glycosylated proteins, they are also secreted and they engage in a cycle that involves calreticulin and calnexin, and they can only live this cycle. It's a quality control cycle. They are their folding is very tightly controlled. They can only be released from this cycle and move along the secretory pathway once they are correctly folded. So we expected this set of proteins to also not exhibit an ER anticorrelation, even more than the other secreted proteins that are not N-glycosylated. And actually, what we observed, to our surprise, we didn't expect that, was a positive ER correlation. Okay, so remember, it's kind of an universal that highly expressed proteins are highly constrained. And then for some authors, it's the only determinant or the most important determinant of rates of protein evolution. But we found a group of proteins with a positive correlation. We don't know why. Maybe there's some mechanisms that promote this positive correlation that in the other proteins, they are masked by these mechanisms that we have been discussed. But the thing is that they escape the ER anticorrelation even more than the other secreted proteins. This is data for different tissues again, and in every tissue, the ER anticorrelation was positive. So in this case, it's not an anticorrelation, it's a correlation. The ER correlation is positive among N-glycosylated proteins. So this is the second conclusion. N-glycosylated proteins exhibit a positive ER correlation. So as I have been saying, for some authors, that's pretty much the only important factor. Expression levels are the only factor affecting rates of evolution. But our analysis don't agree with that view. And then I'm going to continue to talk about different factors that some of them we discovered, some of them had been already mentioned but called into question, and then we have helped establish them as factors affecting rates of protein evolution. So now I'm going to talk about gene essentiality. So what's gene essentiality? This is part of a genome, and this represents the organism that carries that genome. So some genes, if you knock them out or they mutate, the organism still is functional and it can reproduce. Okay, and that's what we call these are the non-essential genes. And you might be surprised to know how many genes are non-essential, at least under lab conditions. For instance, in mouse, at least I have in mind some early numbers that suggested that 55% of genes are non-essential in mouse, at least in the lab. Maybe in nature they are essential. And other genes are essential. If you knock them out, then the organism is not happy, it can't exist or reproduce. Okay. So Hurst and Hirst in 1999, they tested whether essential and non-essential genes, they evolved at different rates. And for that, they used a list of mouse essential genes. And for each of these genes, they found the ortholog in rat, and they calculated the NDS. And then they compared both groups of genes. And that's what they found. These are essential genes and non-essential genes. So non-essential genes, on average, do evolve faster than essential genes. However, they also realized the fact that an important fraction of non-essential genes were immune genes here. And these are known to evolve fast. So they repeated the analysis, removing immune genes. And then they found little of a difference, not much of a difference between essential genes and then non-essential genes once immune genes are removed, okay? So the prevailing idea was that essentiality wasn't a very important factor, or even maybe a factor at all affecting rates of evolution, which was surprising. And of course, there's been more research ever since. And some of the problems of these kinds of analysis are listed in this slide. First, essentiality is measured in the lab. Basically, these genes are knocked out in the lab, and scientists see if the organism still can live and reproduce or not. But in nature, things are different, right? And then the NDS ratios that you estimate by comparing different species, they actually capture the evolution of organisms in nature, not in the lab. When you compare human and rat, we're talking millions of years of evolution, and almost all of these years have been in nature, not in the lab. Another problem is that essentiality can change during evolution. Say the genes that are essential in mouse are not necessarily exactly the same genes that are essential in rat, because throughout evolution, this might have changed. And another problem is that a non-synonymous mutation is not the same as a gene knockout. It's not the same to remove a whole gene than to change an amino acid or a nucleotide that then affects an amino acid. So we conducted experiments. We did an experimental evolution in the lab using Ecology. And by doing so, we are fixing or alleviating at least these two problems. First, now we are studying evolution in the lab, same as, and also rates of protein evolution corresponds to mutations accumulated in the lab. And also we're talking about less than a year of evolution. So essentiality is unlikely to change during that time. We're not talking millions of years now. So what we did was to start this. This is my failed attempt to represent a petri dish. Sorry, I apologize about that. And these are colonies. These are E. coli colonies. And these E. coli colonies already were from a hypermutant strain, so that we could accelerate evolution, so that we could see more mutations. And then what was done in the lab was to every day, someone would pick one of these colonies and then would use it to start a fresh petri dish. And then next day the same and the same. And that was done during 260 passages on the one hand, and then in parallel line B for another 250 passages. And then every few passages, they would freeze, they would get a sample and they would freeze it, that a thing that they called a fossil record that they had in the freezer. And of course, they also froze this starting one. And then as well, every number of generations, they would sequence the whole genome. So we had the sequence of that genome and also the terminal ones, and then also some in the middle. So we could compare these genomes and find out when in the history of these two lines, the different mutations appeared. This is how mutations accumulated over time. One corresponds to, I think these are the non-synonymous and these are the synonymous ones. And this is line A and line B, okay? And then line B evolved faster because we think it's because some repair genes were mutated in the process. And in line B, enough mutations were accumulated to see a very significant difference between essential and non-essential genes. Line A evolved a bit less, and we still have differences, but the differences are not significant. But what this shows is that essential genes do evolve slowly. These are the essential genes in black. Okay. So the third conclusion is that essential genes do evolve slower than non-essential genes. Okay, the next factor that I'm gonna talk to you about is chaperones. Chaperones are proteins that help misfolded proteins or unfolded proteins, proteins that haven't folded correctly just yet, to fold correctly. And then they are interesting. So this is a misfolded protein, and this is a protein whose folding is helped by these chaperones. Then the idea is that these chaperones, they can compensate for certain non-synonymous mutations in these genes. So say, imagine this gene here has suffered a certain mutation that in the absence of chaperones would mean that the protein cannot fold correctly. But the chaperone in some circumstances can help the protein fold despite this non-synonymous mutation. So the idea is that chaperones, by doing that, they would allow faster evolution and even down the line adaptation. It's what we call a buffering mechanism. It allows for more mutations to accumulate. And some results supporting this hypothesis have been published during the years, some by my collaborators, and but these results focus typically on a specific chaperone and in a specific organism, more often than not bacteria, but not only bacteria. And also there were some analyses suggesting the opposite. Actually, on average, protein chaperone clients, I forgot to say, these proteins that are folded with the help of chaperones, we call them chaperone clients. And only a fraction of these proteins are the chaperone clients. Other proteins, the other proteins are considered non-clients, okay? So in some analysis, the clients actually evolve more slowly than the non-clients. So there was some evidence also for the opposite of this hypothesis. The problem, one of the problems is that clients are also more highly connected in protein-protein interaction networks, often more highly expressed, and so on. And these are things that slow protein evolution. So you need to, in each of these analyses, you always have to control for other factors that might be confounding your analysis. So what we did was to, we used this data set in which they looked at the interactors or the clients of 35 chaperones, and then also the data set contained data for 29 co-chaperones. And then for each of these are the different chaperones, and then you have here in color codes how many approximate interactors had been found for each of them. So this essentially, not as represented here, but the underlying data is a network of interactions. So each of these chaperones with which proteins it interacts and with which proteins it doesn't. So from this data set, we derived a list of 4,209 chaperone clients, proteins folded with the help of chaperones, and the rest were non-clients. And we compared their rates of evolution. Sorry, I forgot to say, this is in yeast. And we compared the rates of evolution. And clients, on average, they evolve more slowly. Okay, this is the most relevant graph with DNDS. And also, if you look at DN, it's the same. If we control for expression level or other factors, we still saw that clients evolve more slowly. But when we control for the number of protein-protein interactions, we will see later that protein interactions also slow rates of protein evolution. Then we saw that actually for every category of interactions, chaperone clients actually evolve faster, okay? So for instance, these are clients with a number of interaction between 0 and 5, and this is their median rate of evolution. And these are the non-clients with the same range of number of interactions, and they have a lower median rate of evolution. And this happens for every group. So this is another group, this is another group, and so on. Okay, so that graph suggests that actually chaperone dependence or chaperones accelerate rate of evolution. To confirm this analysis, we did a number of multivariate analysis. I'm going to show you only a couple of them. Here, this is a multiple linear regression analysis, and chaperone dependence is one of the predictors that we use, and see how it has a positive effect on the DN-DS ratio and on DN as well. So that shows that the chaperones accelerate rates of protein evolution. We also did a separate analysis for each of the chaperone groups. These chaperones can be classified into different groups that you have here and here, sorry, and see how HSP70 and HSP90 are the two groups that are probably driving this positive effect, are the ones that have a significant positive effect on rates of protein evolution. The others have no significant effect. So the next conclusion is that chaperones accelerate protein evolution, at least these two groups of chaperones. The next factor I'm going to talk to you about is disulfide bonds. Disulfide bonds are present in some secreted proteins, and they are links between two cysteines in the protein. They can be in the same chain or in different chains, as is the case of insulin, for instance. Insulin has both kinds. So here we have this representation of these disulfide bonds. And disulfide bonds are interesting because they are, in a way, constraining and anchoring the structure of the protein. And then our hypothesis was that if a protein contains these disulfide bonds, then its overall structure depends a bit less on the actual sequence. Okay, so maybe you can change some of these amino acids and the protein can still fold and function thanks to the anchoring effect of these disulfide bonds. So we expected that that would be yet another buffering mechanism similar to chaperones that would also accelerate rates of protein evolution. And this is what we observed. These are intracellular proteins, and then these are membrane proteins, which we divided in proteins with disulfide bonds and proteins without, and see how these evolve significantly faster. And the same happened for extracellular proteins. Again, those with disulfide bonds evolve significantly faster. We control for a number of factors, for instance, controlling for codon adaptation index, which can be seen as a proxy of expression levels. We still see that for every group of for every quartile of codon adaptation index, still proteins with disulfide bonds evolve faster. And here we did something similar, but controlling for protein length. And very clearly, those with disulfide bonds every time evolve faster. And we conducted, again, a number of multivariate analyses. I'm showing you only a couple of them. And all of them confirm that disulfide bonds indeed accelerated rates of evolution or had an independent effect on rates of evolution. Here, disulfide bonds, we included all of these analysis, all of these factors in the analysis. This is again a multiple linear regression analysis, and see how the effect was positive and actually one of the largest ones, together with codon adaptation index. They were and also duplicability. The ones, sorry, and some others as well. But it was one of the ones with the strongest effects on rates of protein evolution. And then this is an ANCOVA, and in this case, it's more clear in this analysis that the presence of disulfide bonds was the factor potentially with the strongest effect on rates of protein evolution, at least according to this analysis. Okay, so the next conclusion is that disulfide bonds accelerate rates of protein evolution. And then the last factor that I'm going to talk to you about is the number of protein-protein interactions. In my lab, and even before my lab existed, even since my PhD, I've been very interested in understanding the evolution of pathways and networks, the evolution of proteins as systems of different proteins. And we've done quite some work on signal transduction pathways and showed that they, in terms of rates of evolution, they evolve differently from metabolic pathways. In signal transduction pathways, often downstream proteins tend to be more the ones most constrained, whereas in metabolic pathways, it tends to be the opposite. And also, we have studied more larger scale networks, such as the protein-protein interaction network, and not so much the metabolic network, but they are different. I show them to you in this slide because they are different. In a protein-protein interaction network, each of these nodes is a protein, and each of these lines represents a physical interaction between these two proteins. These two proteins, they have been shown experimentally to interact physically at some point. Whereas in a metabolic network, what we tend to have is metabolites that are, they are parts such as here, for instance, that would be an example. Here we have the different metabolites, and then each enzyme uses one as a substrate and produces a product that then is a substrate for the following one. Okay, so in this case, these lines don't represent necessarily a physical interaction between the proteins. So we have worked quite a bit with these kinds of protein-protein interaction networks. And then one of the things that had been shown, and we also found in other species, is that in general, there is a correlation between the number of protein-protein interactions and also other measures of network centrality and rates of evolution. So let's say, for instance, in this network, you would expect that this protein that has many interactions would probably evolve slower than this one here that only has one interaction. At least on average, that's what we expect. And this had been shown. Here we have data for Arabidopsis thaliana. This is the Arabidopsis thaliana protein-protein interaction network. And here, this is not the number of interactions. It's betweenness, which is another measure of network centrality, but the idea is kind of the same. And you see a negative correlation, but it's not a very clear negative correlation. For some authors, actually, interactions, the number of protein-protein interactions had only a minor effect or maybe even no effect. And then this correlation, maybe it was thought that it could be a byproduct of highly connected proteins being also highly expressed, or also because the network, the protein-protein interaction data is in general poor quality. There's a lot of false positives, there's a lot of false negatives, there's a lot of biases in the data set. So some scientists thought that actually the number of protein-protein interactions had no effect or only a marginal one. So what we did was to study the human protein-protein interaction network, which we obtained from the BioGrid database, a total of 213,000 interactions. And we also studied a signal transduction network that had been manually assembled by Cui and collaborators. In this is only a part of it. In total, there were over 1,500 proteins here. I don't think they have represented all of them. But the idea is that each of these interactions, it has been confirmed by looking at the literature and by multiple authors independently that actually the two proteins interact. Some of these protein interactions are physical, some are not physical in this data set, but the idea is that this data set is of much higher quality than this data set, which comes from many different experiments. Many of them are large scale and produce thousands of interactions at a time. So we found that the number of physical interactions and also other metrics of network centrality did correlate with rates of protein evolution, but also so did other factors. So we have to control for these factors. And then one of the methods that we used for that was the principal component regression analysis. In this graph, each of these bars represents one principal component, and then the different colors represent what this component is made of, what is this composed of, okay? And then I represented with this, or in these black boxes, the factors that had to do with network centrality, essentially number of protein-protein interactions, betweenness, and closeness, different measures, and see how they do account for a significant fraction of the variability explained by each. So I forgot to say how high each of these bars is represents how much of the variability of rates of protein evolution, in this case, are explained by each of the principal components. And see how, in this case, the network centrality has an impact that is comparable to the measures of gene expression, okay? Which is something that earlier studies has suggested that wasn't the case. Earlier studies using similar statistical analysis, they suggested that pretty much expression was the only factor affecting rates of evolution. So we found that not to be the case. And similar results were obtained when we used the high quality network, this manually curated network I was telling you about. And also similar results were found when we studied other organisms, like yeast, Drosophila melanogaster, and to a lesser extent, C. elegans. So the last conclusion of this talk is that protein-protein interactions do slow protein evolution. And I finished by thanking my collaborators that participated in these studies, and my funding sources, and of course you for your attention. So if you have any questions.